I'm going to show you here an example of using Plex to do spatial transcriptomics. And we're going to use an example, a really nice data set from Stec et al. From Regeneron, looking at DMD, so Duchenne Muscular Dystrophy, specifically this D2-MDX mouse model, really nice spatial transcriptomics data set. We're going to go and look at this portion here where they looked at specifically these type IIb fibers in the muscle and looked at disease models, so MDX model compared to wild type genes that were up and down. They saw, for example, a lot of genes involved in wound healing and some other things. So we're going to take that gene list and put it into Plex and see what more we can find. And so I've got here the supplementary file. You can see I have it on the Type IIb filter tab here. They've got data for the other fibers, and there's other cell types, and there's other data as well in this paper. But let me copy this gene list here, these gene symbols, just a Control-C copy. These are the genes that are upregulated. And we're going to come over to Plex. We're going to paste them in. And those are going to map initially to human genes. And we're going to also change the setting here to mouse and paste them in again. So we've got both the human and the mouse versions of these genes. And we're going to run that search. The search takes about 20 to 30 seconds. So to not waste any time, I've got it pre-run here. So let's take a look at the results. And we're going here directly to the RNA profiles result. This is other independent studies. So we've integrated all the major gene expression databases, Geo, Expression Atlas, MSigDB, and others. We're able to see where have we seen a gene that's like this before. So where has this kind of profile been seen? And you can see here out of about over 11,000 RNA profiles, the very top hit here is another study, independent study, also looking at MDX model of DMD, looking at skeletal model. In this case, they're looking at the CMAH knockout effect. So we're linking it to another study, clearly very relevant, the same mouse model, a slightly different take on it, focusing in on CMAH. And as you go down the list here, you see that there's other studies that are also clearly relevant. This second one around skeletal muscle regeneration, chondrocyte differentiation. As you go down, hind limb muscle comparison, wild type, again to MDX mice. So there are, so that gives you some confidence that this gene list is really reproducible. We see this in independent studies. You can see here, there's 101 genes that are in the overlap, for example, with our list and this particular model. You can click on any of these results and see what are the actual genes that are modulated. In many cases, also, what are the levels of modulation? So we can also really zoom in and focus in on the genes that are driving these connections. So, and here you see some of those 100 genes that are linking these two studies. So this is a great place for hypothesis generation about what are the specific contexts and maybe the specific modulators and disease processes and pathways, et cetera, that can give rise to this gene signature that we're seeing from this, specifically these type 2 fibers in this DMD mouse model. So there's a lot on this page. I don't have time to really go through it all here. The top, probably 100 results are really pretty clearly relevant, and there's some interesting things in there. Again, I don't have time to go into too much detail. Let's switch over to some of the other data types. So we come over, for example, to proteomic profiles. We can look to see, are there compounds that modulate the levels of these genes, or at least many of them, at the protein level. So the proteomic profiles here, this is the MOA Atlas data from the Steve Gigi lab. These are proteomic profiles in response to compound treatments. And so we can see which compounds partially phenocopy this type 2b fiber transcriptome response, in these mouse models. And what we can do, because we put in the upregulated genes, we can also ask, are there compounds that reverse the disease process, that push the genes in the opposite direction, in this case at the protein level. And what we can see is that there's some interesting results at the top. We've got a proteasome inhibitor that apparently downregulates 10 of the genes. You can see the number here, 10 of the genes that were upregulated in the MDX mouse. Proteasome inhibitors are known to have, have been reported to have anti-fibrotic effects. We see baphlomycin A also having downregulating some of these genes. This is an autophagy inhibitor. Autophagy is also clearly linked to fibrosis. And then the third hit is a TGF-beta receptor modulator, which is a known part of the fibrosis pathway. So we really, there's some interesting top hits here. The overlap is a lot less than what we saw with the transcriptional profiles, which is partly to be expected just at least from the data type change. These are protein levels and our input data set were RNA levels. So that could explain this in part, but this is an interesting hypothesis generation about potential pharmacological interventions that could affect this disease process. If we move over to perturb-seq data, we can see that we've got genes that are modulating, so knockdown of these genes, and actually let me remove the downregulated. So the genes that are upregulated are similar. There's some overlap. 38 of these genes are also upregulated by the knockdown of CHMP6, which is involved in vesicular sorting, endosomes, lysosomes. So there potentially some link to what we just saw maybe with baphlomycin. But here we're looking at the genetic level. So this is CRISPR knockdown of these genes, a full RNA-seq profile of each gene knockdown. This is data coming from the perturb-seq data from the Weissman lab. So this gives us some ideas about genetic modulators that may lead to the profile that we see. You can see that there's a number of genes here, and potentially by looking at this gene list of genes whose knockdown phenocopies or partially phenocopies the MDX mouse 2B fibers, that might give us some insight into what pathways are actually involved in that process. So these genes here could be of interest as potential modulators of the process. And so again, something to generate hypotheses, to look for potential convergence with data that we're seeing in these other data categories, the proteome profiles, the RNA profiles. And so finally, let's come over and take a quick look at CRISPR screens. And here we're asking, are these genes, any of these genes, hits in a CRISPR screen? And this is from the ORCS database. So for example, we have regulators of phagocytosis. So some of these genes, when you knock them down, modulate phagocytosis. I'm going to scroll down just a little further because we see there's also this link to this autophagosome process, that a number of these genes are hits in that screen. And for any of these, and this is the case for any of these data categories, you can click on the link, brings you to the original source of the data. In this case, it's the ORCS database in this particular CRISPR screen. So you can see what, the hits that we're seeing here, how strong were the hits, what was the screen that was run, how were the hits called, et cetera. So it's really completely transparent results. So you know where all the data is coming from, and you can bring that into your interpretation. So with that, I'm going to stop there. There's really a lot more in this data set, and there's a lot more to dig into. Obviously, we've looked at just one of those data points. So the type IIb fibers, there's other gene lists we can put in here and other data in this study. We've identified other studies that are also relevant. So these are other studies that we could dig into also in a similar way, and really pick apart the pathways that are involved in the disease process, identify potential modulators both at the pharmacological level and kind of this genetic approach with CRISPR, and really get some insights, potentially some new insights into what these disease processes are with a really high level of resolution. So if you found this interesting, please reach out. Please feel free to contact me. My name is Doug Selinger. I'm the founder and CEO of Plex Research. Check out our website as well, at plexresearch.com. And I'd be happy to discuss this with you in more detail. So anyway, I hope you found that interesting, and I thank you for your attention.